Right. Okay. So our recording is already ongoing.
Thank you everyone for joining. We will just wait for a few minutes for others to join and we will start the meeting sharp at 3:05. Hello everyone, good afternoon. I'm delighted to welcome you all to today's webinar on next gen sequencing hosted by Christian Biosystems. Myself, Annie Florence, I'm the senior application support working at Christian Biosystems. So just to talk about Christian Biosystems in brief, we are based in Mumbai and it was founded in 2003 and we are a biotech firm providing quality kits, reagents and consumables to scientists to support their research. We have a strong background and expertise in immunology, and we work with biopharma, CROs, and vaccine and academia at pan India level. To introduce Rapid Novor, it's based in Canada, and they provide world class de novo sequencing and antibody characterization services. They have expertise in proteomics, and they have completed over 800 plus projects successfully. And in today's meeting, I would request everyone to, expect, learn, and have some insights regarding the next generation protein sequencing. And I would also like to introduce Monique, our senior scientific sales executive from Rapid Novor, who has joined with us, today's speaker. And she has worked collaboratively with hundreds of leading scientists in both therapeutic and diagnostic fields globally to understand their goals and apply her proteomics knowledge in tandem with Rapid Nova services. Throughout the session, if you have any queries, if you have any questions, you can always drop a message, and the same will be addressed during the Q&A session. And I would request Monique to take over the meeting and continue with the presentation. And one more, if you have anything to speak about, if you're having any difficulties, you can always drop a message. And we request you to stay everyone on mute. Thank you. Over to Monique.
Thank you so much, Annie, for the introduction. Let me just get my screen up for you here. All right. So, as Annie mentioned, my name is Monique Seymour. I'm a senior scientific sales exec at Rapid Novor. The company is a Canadian company. The accent that you're hearing is a Canadian one, but I'm personally based in London, UK, as I work with our partners and clients on the other side of the world. So today I'll be talking to you about next generation protein sequencing, as well as characterization for antibody research and development. So as an overall agenda, just to quickly run through it, I'll do a quick introduction into Rapid Novor, and then I'll get into detail about our protein sequencing services, REMAB, as well as REPAB. And then I will chat about our introductory, an introduction to our characterization services, and I'll be providing some user cases for all of these different applications throughout the whole presentation. So a bit of background on Rapid Novor. We are a Canadian proteomics company based in Canada. We are the largest privately funded mass spec facility in Canada. We were founded in 2015, and I don't want to read all the all the things I've listed on this slide, but we do have a lot of expertise in this field. We are a world-leading company within next generation protein sequencing, and we are constantly innovating and improving our techniques and methods for a variety of different applications within the industry. So we're very lucky to have the scientific team that we have on board to help lead us in the right direction. So one of Rapid Novor's philosophies is that we want to shift antibody R&D by starting it at the end. So you're starting with antibodies that you know already bind, and then you want to analyze and engineer and improve them. And the way, or one of the ways in which we do this, is through next generation protein sequencing. And now I've highlighted that P there in orange because it's not to be confused with next generation sequencing. Although it sounds similar, it is a very, very different technique, which I will be explaining about in more detail. So just as a quick high-level overview, these are a few of the different proteomic services that we offer. As I mentioned, I'll be discussing our REMAB and REPAB protein sequencing services, and then I will also discuss our epitope mapping and SPR services for your reference later today. Now, I know that we have a variety of different scientists that are attending this webinar, all from different specialties and teams and workflows, I'm sure. So I just wanted to call out and list a few different teams that we work with and how our services can apply to you. So I have tried to provide general use cases that hopefully somewhat closely align with what you're potentially working on. But I definitely can't promise that I'm being able to, work across the board and call out every single one of them. So, if you are working with antibodies and proteins, then there is a very high chance that we'd be able to help you in some way or another. And that's typically what our clientele have been. So to focus in on our next generation protein sequencing solutions, I'd first just like to give you a very high level overview of what that workflow actually looks like. So typically, we're starting with a purified antibody. We then are going to process it with some digestions and treatments. We'll run that through our mass spec, and then we'll actually do the sequencing work and provide that within a final report for you that details that antibody or protein sequence. Now, specifically, if I want to call out, how Rapid Novor is able to enhance this service, we have a few different techniques that we do within our process, which allow us to get the really high end results that we are able to. So we actually use between 5 to 10 different enzymes, or we complete 5 to 10 different enzymatic digestions to do and prep the protein for the mass spec. We have a variety of proprietary wet lab techniques to treat these proteins to optimize the peptide results that we get from the mass spec. We also are very lucky to have a world-leading machine learning algorithm to complete the next generation protein sequencing. This algorithm was actually written by our CSO and co-founder, Ben Ma. He has to date written 2 out of the 3 existing de novo protein sequencing algorithms that exist, and we use his most up-to-date recent one. And then, of course, we're very lucky to have the bioinformatics specialists that we do on our team. All of these techniques together allow us to obtain end-to-end coverage of the protein. So we're going all the way from N-terminus to C-terminus, covering every single amino acid to a very, very high level of confidence. We are also able to differentiate between isoleucine, which I'll discuss in a bit more detail. And much like our name, we're fast. We get this done within 2 weeks with a very small amount of protein. So I just want to highlight a few of the points that I mentioned there. So what you're looking at is the peptide coverage view for a sequence that we found from our de novo protein sequencing or next generation protein sequencing. So as you can see, and just to break this picture down a little bit, so all the ticks that you see on the very top of the picture there, those are representing amino acids. The green and orange region represents the variable region. The specific orange regions there represent the CDRs. And then the blue that's cut off would be the constant region. And then all of the different colored overlapping lines that you see are different peptides that we obtained from our mass spec to build the confidence in what the sequence is. So, when you have peptide coverage like this, there's, absolutely no doubt. It's as high as you can statistically get to, 100% confidence in the sequence that we're obtaining. And I would say, the specific coverage map has been cut off. It's a very, very high level one. You don't need this high level to get that 99.9% coverage. And, but we do aim to get as many as possible because we believe that the quality of the results is very important for further downstream activities. We also can do isoleucine-leucine differentiation. So for those of you that aren't quite familiar with the difficulties here, isoleucine and leucine do have the same molecular weight. So when running them through a mass spec, it can be difficult to differentiate between the two. And what we've done is we've developed this technology that we call WILD. It's our fun little name for W-ion isoleucine-leucine differentiation. And basically what we can do is we can break apart leucine and leucine from their side chains, and the resulting ions, the W ions, have different molecular weights. And so when we're looking at the mass spec, we can differentially tell, which one is a leucine and which one is an isoleucine. And this is very important because on average with antibodies, you can get between 2 to 6 different isoleucines or leucines within a CDR or variable region of an antibody. And of course, these are very key, important areas that you don't want to be second guessing and can, make or break the activity of your antibody. In terms of requirements and timelines, so ask for 50 micrograms of purified antibody. We can work with any species and any isotype. We can work with conjugated antibodies as long as the conjugation is not site-specific. Any buffer is fine. We like to work with pure samples because we are working with mass spec and we want to limit the amount of background protein noise. And we don't have any lead time. So as soon as we receive a sample, we can get started within just 2 weeks. Now I'm going to just quickly describe a specific case from one of our clients that used REMAB specifically for protein engineering. So the DeLuca lab at Colorado State University, they needed some antibody fragments to use for their assays. They study mitosis and were ultimately able to bind kinetochores in mycotic cells and see immunofluorescence. So they started with a small sample of MAB protein, which they sent to us. And then, of course, we completed REMAB protein sequencing, provided them with a full antibody sequence. And with the understanding of that sequence, they were then able to do species swapping and engineering of the antibody fragments. And because they had very high confidence within that sequence, they were able to get a sensitive and reproducible end result, which was key for their work. This is a different use case in which REMAB was used for the use in an immunoassay development. So Dr. Wang from the Zubarov lab was actually previously guested on one of the webinars that Rapid Novor previously did a few months back. And she presented her work on an immunoassay for Alzheimer's disease. So in this specific study, isoaspartate accumulation in human serum albumin was used as a biomarker for the detection of Alzheimer's. And she actually had a set of isoaspartate specific monoclonal antibodies, which were generated in hybridoma cells. However, 2 that were of interest had difference of just 1 amino acid between them. And so it was very important to be able to differentiate which was which in order to move forward with one of the antibodies. And so although she had the genomic sequence, it was important to then confirm that to a much higher degree of confidence using next generation protein sequencing, or also known as de novo protein sequencing, using REMAB. And so we completed that work for her. And with that sequence, she was then able to move forward with the IVD development and preclinical testing of the specific assay. Now, the next service that I want to discuss with you is REPAB. Now, REPAB is our polyclonal antibody sequencing service that uses next generation protein sequencing. So there's a lot of content on this slide, so I will break it down quite slowly. The first image on top there is what you see is the end-to-end workflow of what we can provide. Of course, REMAB de novo polyclonal sequencing is the very specific next generation protein sequencing service that we offer. And if you look at the picture at the bottom, it does look quite similar as REMAB did, in that we are doing multi-enzyme digestions, we're running that through the mass spec, and then we're assembling all that data. However, I will say it's a completely different beast of a project. Polyclonal sequencing, as you can imagine, is much more complex due to the extreme diversity of the samples that we're looking at. So this will take much closer to 3 months' time, and we also ask for much more sample. So ideally, the starting material is 1 milligram of a single source antigen affinity purified IgG. And just provide you with some context on that full process that you're seeing in the first picture there. So, of course, we can organize this end-to-end process for you. The immunization we don't do in-house. The PAB purification and testing we can do in-house. Of course, REPAB polyclonal sequencing is our very unique expertise that we offer here. And then we can outsource the recombinant expression, and then we can do some of the characterization work in-house, which I will discuss in some slides later on. Yeah, it is an interesting application that is very unique to the market. It does offer a completely different way of doing antibody discovery from the standard protocols, and it's a in my mind a fun way of doing or I should say applying next generation protein sequencing in a way that was completely unthought of before. Really being able to look at and select from a native immune response of the circulating antibodies within an animal or a patient. So it provides a completely different perspective, and it also allows you to work with antibodies that you want to work with instead of whittling it down from the total antibody within a species. You can specifically sequence ones that are to your target, that are functioning the way that you want. So yeah, it's a very unique capability. So this is one example of our polyclonal sequencing workflow, in which we had a rabbit that was immunized with a peptide. We then obtained polyclonal serum from this rabbit that was purified, antigen purified as well as IgG purified. And then we were able to de novo protein sequence or next generation protein sequence the polyclonal antibody to obtain full length monoclonal antibody sequences. So this is an example of some of the coverage that we got over the heavy chains and light chains of one of the antibodies that we found. So as you can see, this is excellent coverage over this antibody. Every single amino acid is covered over many different peptides that we see there. So we're very confident in the sequences here. And in total, from this polyclonal, we were able to sequence 13 different antibodies. Of that, we decided to express 9 for recombinant expression. And when we tested the 9 via ELISA, we found that 7 of the 9 bound very similarly to the original polyclonal antibody. So that polyclonal antibody is just in that gold brown color there. And you can see, the polyclonal antibody activity is going to be the average of all of the different monoclonals that you would find within that PAB. So that's when you look at this picture, it makes sense that that polyclonal activity or binding is where it is, because you can get an idea of, okay, from all the activity of the 7 that were expressed and bound, it does look like it would be the average accumulation of that activity there. So next, I'd like to share our characterization services with you. So specifically, we offer HDX MS epitope mapping. Again, this is using mass spec. And then we also offer SPR kinetic analysis. And these are a couple of the services that we would offer as a downstream workflow to that overall discovery workflow I mentioned earlier. After you were, say, to express antibodies and you want to understand a bit more how your antibodies were acting and where they were binding, and maybe select a monoclonal antibody that you liked most for your application. So first off, for SPR kinetic analysis, this might be a workflow that you're familiar with doing. But generally, how it works is we have a ligand and an analyte. We'll immobilize that ligand onto a sensor chip. We will then inject the sample, and generally we've optimized the conditions to make sure that we have the best possible binding results here. So we will inject that analyte and then wash it off, and then we're able to get an idea of the binding kinetics of that analyte and ligand. Often this is a antigen antibody interaction. The unique advantages from what we do is that, it is real-time monitoring. It can be label-free detection. We do not require a large amount of sample. So, over 100 micrograms should be fine. And we like standard buffers, so we don't need anything fancy or any detergents or something like that to be able to accurately view the kinetic output of the interaction. Next up, we have our HDX MS epitope mapping. So I would say this is more likely to be an application that perhaps you're not as familiar with. It is more niche, but there are quite a few um, enhanced pieces of information that you get from using HDXMS for epitope mapping. So the overall workflow I'll quickly describe is typically you're looking at the bound versus unbound states of the protein. Typically, you're going to incubate your different fractions of your protein with D2O or deuterium, and these will readily exchange with hydrogens found on the protein. So through quenching, and by quenching, you're controlling the temperature and pH and preventing back exchange of exposed hydrogen atoms with deuterium. You can do this with the bound and unbound state. And if you're looking at the bound complex, then in theory, you would have areas in which there are less deuterium uptake because they are involved with the binding of that antibody and they're no longer free for that exchange. So we digest that complex, we get the peptides, and then through mass spec, we're able to look at the differing molecular weights to differentiate between, what areas of that protein took or had deuterium exchange, and which ones potentially had less deuterium uptake after they were bound. I highlighted a key benefit here in that we can identify linear, conformational, and structural epitopes. So it is very unique because sometimes over a period of time, you could have proteins that, get comfortable being bound together. And perhaps the conformation of that protein changes a little bit and a specific area of that protein then opens up and is exposed, or perhaps it's the opposite and it does get less exposed. So you can get an idea of that. We are doing this at 3 different time points. Generally, it's, at 2 minutes, 10 minutes, and 30 minutes, but this can be optimized for your specific protein. And so you do get some really interesting data, not just of, a potential area for binding, but down to the peptide level of, 3 or 4 amino acids over a period of time. It's I think it's really key information there. In terms of applications, typically it is antibody antigen that we most often work with. But of course, if you're looking to do some patent filing, it's a great way to, it's a great analysis to have included there. We can also compare biosimilars. We can look at batch to batch variations. We can look at stability. So yeah, quite a few different applications of the specific technique. So now I have one case study that walks through, our protein sequencing for polyclonal antibodies all the way down to how we can better characterize and potentially select for specific antibodies. So in this specific case, we had a camelid that was immunized with a rabbit polyclonal. We were then able to do antigen purification. Once we had that PAB, we of course did the digestions and ran it through the mass spec. And one thing that I didn't mention previously is that we can optionally also take a look at NGS data just as a supplementary piece of information to use. The method is proteomics led, of course, but NGS data can just be a nice extra piece of information to have. Now the output for this was that we identified 10 different sequences. From that pool, we decided to express 6 for synthesis and characterization. And what we wanted to do was look at how it was binding and where it was binding, because we wanted to potentially select for, unique antibodies that would be useful for, different purposes. Now, as I mentioned, the antigen in this case was a rabbit polyclonal. So we selected two specific rabbit antibodies, so P17 and CD3E epsilon. And those two antibodies were used as the antigen to test the alpaca mAbs that we found from the alpaca polyclonal that we sequenced. So when we tested this against P17 and CD3 epsilon, we of course found unique antibodies there. So interestingly, you are going to get different affinities because, of course, these antibodies are binding to different antibodies, differing antigens, I should say, because that antigen was polyclonal. So in this case, we can see that R9 is consistently a top binder with a very high stability and high affinity. Interestingly, R7, although it's a very high stability, high affinity binder for P17, it had absolutely no interest in binding with CD3 epsilon. And then, of course, R4 is quite consistent in its affinity, quite an average affinity. And R1 was a very weak binder. And then, of course, we have R2 and 10 that were non-binders to these specific mAbs. So that's not to say that if we brought in a different, rabbit antibody, that we would, of course, had very different results, right? So these are just different ways in which you can obtain diversity in your monoclonal antibodies, and also, select for monoclonal antibodies that fit your specific criteria. So if you were looking for a very highly stable antibody, but had an average affinity across the board, then maybe R4 would be a top pick for you. And now just looking at some isoaffinity plots, so doing comparing the affinity of the antibodies that we found to P17 and CD3, you can more clearly see visually which antibodies might be of most interest to you. We also decided to do a binning analysis here, and that's using our SPR capabilities. So in this specific case, with epitope binning, we're looking at the differences of, the order of antibodies here. So the saturating antibody would have been the antibody that bound first to P17, and then the analyte was run after that saturating antibody had been bound. So as you can see, R7 and R9 are quite happy to be bound second, but R4 really was not happy to be bound if it was the second one on P17. And we were curious about this, so we decided to do epitope mapping. As a first step in epitope mapping, we do a sequence coverage of the heavy chain and the light chain. And then, of course, we do the deuterium exchange, we obtain the results, and the output from that deuterium exchange is a heat map in which we can observe the relative uptake of deuterium. And if we're focusing on amino acids 275 to 282 there, we can see that there was a decrease in deuterium uptake when looking at one of the antibodies to R9. So this is just one visual, right? So we would have had 4 of these. And this is specifically for the heavy chains. We'd also have ones for the light chain, but this is just one visual that you would get in a report for HDXMS. And when we used HDXMS to then plot the results onto the structure of the protein, things start to make a bit of sense. So we can see here, R9 has, one unique binding site, so that makes sense. So does R7. But if we look at R4, we can see that it requires 2 different areas of the antibody in order to bind to P17. And so it makes sense that if there was anything else binding in those areas on different locations, then it wouldn't have been able to successfully bind. So yeah, really, really interesting insights. Very useful if you wanted to have different epitopes or have antibodies that are binding to different epitopes in a specific assay. This is definitely good information to have and understand how they could be interacting with each other or canceling out binding of each other. And then, of course, we can obtain key insights from, that integrative approach of using SPR and HDX and highlighting, potentially if you wanted an antibody with a lower affinity or a highest affinity or a specific epitope at the Fc region. Yeah, it can be a very unique way to get fit for purpose antibodies, depending on what your um, end goal is. And of course, we're finding this from a native immune response. So there are definitely benefits in working with antibodies that have already been biologically, naturally selected for by Mother Nature. So I've highlighted quite a few different services and mentioned a few different use cases, but I just want to quickly show how these applications could be used within a general, discovery process. We work with clients in a variety of different areas of a workflow, depending on, when you do what analysis. So some oftentimes, you want to do epitope mapping very early on and just automatically get that key epitope that you want to be working with. And other times you maybe have 2 or 3 key antibodies that you are wanting to select from, and then you do epitope mapping. So that choice is of course yours as to how you want to find that antibody. And then specifically for protein engineering, I did mention protein engineering a few times throughout the presentation, but I of course want to call out that this is generally the process for when you're designing an antibody, right? You want to understand what you're working with, design and fiddle with the specific areas of interest to potentially optimize binding or affinity or blocking. Then you're going to produce it and you're going to observe what it does, right? So, of course, for understanding that antibody, knowing that sequence, that epitope, the conformation of that antibody when it's bound and potentially the affinity are really key pieces of information, all of which the services that I mentioned, the next generation protein sequencing as well as the characterization are useful for. And then, of course, to observe that, if you wanted to understand and make sure that that protein that you've produced is binding where it should, or if it's been produced correctly and that sequence is correct and that one amino acid change that you made is there, and make sure that there's no post-translational modifications. Again, really key piece of information to confirm that you've produced what you want, or maybe to confirm that, hey, actually, I haven't produced what I want. I need to go back to the drawing board. So that is the end of my presentation. And I will be happy to open up the floor for questions.
So I would request participants if they have questions to drop the messages so one can answer for it. And it's not a question or a query. If you have anything related, you want it to be elaborated a bit more, and only we will just wait for.
Sure, no problem.
Okay, I would repeat, if anyone has any questions to specific slide, you can drop your message. Yeah, we will be emailing the recording to you. So in case afterwards also, if you have any queries, you can come back to us. Right now, if you have any questions to any specific service, or if you're not, if any case study or any technical queries, then you can surely ask right now. Thank you. Okay, so Monica, I guess there's no question. So if there is anything in later after we share the recording, I guess we must be having some queries which we can, later discuss lately. Okay, thank you everyone all for attending the webinar today. We are very grateful for your participation and hope you found the session informative and engaging today. And we appreciate you taking the time out from your busy schedule to join us. And special thanks to Monique for sharing your expertise and insights into protein sequencing. In case after this webinar, we will be sharing you the slides and also the recording sessions. In case if you have any queries, you can always drop us an email and we can take the discussion forward. Thank you, everyone. Thank you. Thanks for joining. Thank you, Monique.
Thank you so much, Annie. Thanks everyone for joining.